Assalamualaikum warahmatullahi wabarakatuh. The vegetative growth response of Detam soybean varieties toward Bacillus subtilis and Trichoderma species applications as bio-fertilizer. Black soybeans are a variant of the soybean plant whose grains are used as raw material for the soy sauce industry. Efforts to increase black soybean production are also part of efforts to increase national soybean production, especially through the use of available marginal dry land. The ability to produce and plant productivity is largely determined by the success of the plant through the critical phase, namely the vegetative phase in the rhizosphere conditions which are biologically unfavorable for plant growth. Efforts to deal with environmental stress in plants are by utilizing effective microbes that can help plants and increase soil fertility. From the Trichoderma species, whose role is to provide protection for plants from diseases in the rhizosphere, from the group of effective bacteria, a number of Bacillus species which also has the ability to produce growth-regulating compounds. It is necessary to reveal the extent of activity and possible interactions between, these two types of microbes that are effective in making a real contribution to plant growth. This study aimed to determine the effect of the bacterial isolate Bacillus subtilis and Trichoderma isolate and its interaction on the growth of the Detam black soybean plants until the end of the vegetative phase. Preparation and planting. Trichoderma isolate and Bacillus subtilis isolate used in this experiment was a collection of the microbiology laboratory of the Muhammadiyah University of Sidoarjo. The soybean used in this experiment was black soybean with Detam 3 Prida, which was released based on the decree of the Minister of Agriculture. The experiment was arranged factorial using a completely randomized design. The first factor was the application the subtilis, which consists of without and with the subtilis, while the second factor was the application of Trichoderma, which consists of without and with Trichoderma. The experiment was repeated 4 times. Each experimental unit consists of 4 polybags containing 4 plants each. The observed variables were plant height, stem diameter, number of leaves, leaf area, stover dry weight, root dry weight, and the population of the subtilis at the end of the observation. Data were analyzed by ANOVA at the 5% level. A treatment mean was compared with the control without the subtilis and without Trichoderma to determine the percentage increase or decrease in control. This is the result. Picture 1. Growth height soybean varieties Detam. Table 1. Average effect of Bacillus and Trichoderma to the height of Detam soybean varieties and percentage increase or decrease compared to control. This is growth of stem diameter soybean varieties. The average effect Bacillus and Trichoderma to the stem diameter and the percentage increase or decrease compared to control. This is growth of the number of leaves of soybean Detam. average effect of Bacillus subtilis and Trichoderma on the number of leaves of Detam soybean, and the percentage increase or decrease compared to control. This is leaf area. This is average effect of Vegetative and Trichoderma on leaf area, and the percentage increase or decrease compared to control. This is average effect Vegetative and Trichoderma to dry wax of stopper and root dry wax of soybean plant, the temperature, and the percentage of increase or decrease compared to control. Table 6, average effect Vegetative and Trichoderma, again, the population density Vegetative. The vegetative growth of black soybean in response to the activity Trichoderma and Vegetative in the reserve showed a very large increase of both twofold compared to the treatment without Vegetative and Trichoderma. Trichoderma used mostly organic matter as substrate for its activities. The composition of organic matter by Trichoderma with the help of very degrading enzymes that is produced will contribute nutrients to meet the need of plant growth, whose response are shown mainly in the form of stopper dry wax and root dry wax. Bacillus were able to increase the average stem diameter, and stopper dry wax and root dry wax respectively 220% and 195% at the end of the observation. This bacteria produce secondary metabolite which is regulator of plant growth. Trichoderma not only acts as a fertilizer supplier because of its ability to degrade organic matter, releasing nutrients for plant needs and helping plant grow. The real interaction between the effect of Trichoderma and B. subtilis on all vegetative growth variables shows a synergy that results in the growth response of the Detam black soybean plant variety in this experiment. Conclusion: the interaction effect of the application Trichoderma and the bacterium Bacillus subtilis isolates was very significant on plant head, stem diameter, number of leaf area, stopper dry wax, and root dry wax of soybean varieties Detam. Subtilis bacteria produce the highest increase in leaf number, stopper dry wax, and plant root dry wax, while the combination of Trichoderma and subtilis produce the highest increase in plant height and leaf area of soybean. Giving subtilis bacteria alone does not show the role of helping plant optimally. The presence of Trichoderma supports the activity of subtilis in the rhizosphere of the Detam soybean plantation. The combination of these two types of effective microbes can be further developed to be formulated into a bio-fertilizer for plant fertilization action. Thank you. Assalamualaikum warahmatullahi wabarakatuh.